message there. Once you post it, she'll be able to reply, and you'll be able to see that there's a reply from the teacher.
And so technically right now, if you guys, me and her just tested it out, so if you go into e-learning right now, you should see the response. I think I said okay, that I sent to Malak. So if you can't see that, let me know. But if you can log on right now just to make sure, that way, when one person has a question, everybody can get the answer to the question. Okay, instead of you guys emailing me directly, you can email through the message. Thank you, Malak. All right, okay, you guys, let's get started. Okay, so we're gonna finish cell cycle today, and then start on cancer biology. I will tell you that you will not be responsible for slides 36 and on for cancer bio, okay? All right, because I don't think we'll be able to get through it. All right, so going back to cell cycle, we talked about the different phases of the cell cycle, the proteins that are important during the different phases, the cyclin D, CDK4, cyclin E, CDK2, cyclin A, CDK2, cyclin B, CDK1. Cyclin A, CDK1 is important during G2 phase. We talked about the fact that these cyclins cycle, so their transcription goes up right before the phase that they're supposed to be functional in, and then they get degraded by the proteasome when you want to exit out of that particular phase of the cell cycle. So another way that the activity of these complexes is inhibited are through these negative regulators of cyclin. So I've got this table here for your information. So we've got one family of cyclin or CDK inhibitors is going to be the cyclin kinase inhibitor family, the ecm4 family. And the proteins that are part of this family are P15 and P16. And what they do is maintain cells in G0 phase. So during G0 phase, these proteins are high, their levels are high, and what they do is they bind to some CDK4 and CDK6, and they sequester CDK4 and CDK6 away from any cyclin D. So that's their claim to fame. They're going to be high in G0, they're going to be binding to cyclin D, CDK4, or 6 so that it cannot bind to cyclin D. And what happens is during, once the cell enters into the cell cycle phase, so enters into G1, these ink family proteins, their levels go down. So now their levels are going down, so they're not able to bind and sequester CDK4 or 6 away from the cyclin D that starts to be produced during G1. Then you've got another cyclin kinase inhibitor family, the CYP-KIP family, so that's P21 and P27. And they're important to prevent blocking the cell cycle if DNA is damaged. Their target is going to be CDK2. So what they do is they'll either bind to the cyclin A CDK2 complex, or they'll bind to the cyclin E CDK2 complex. And so here, what I've got is P27. Its levels are high during G0 phase. And what we'll see in, I think, the next slide is when you treat cells with mitogens, what happens is P27 levels go down. So now you don't have this P27, this inhibitor blocking the CDK2, being able to block that kinase. If the cell experiences any damage, DNA damage, there's a famous protein transcription factor called P53. P53 happens to be mutated in about 50% of cancers. So what P53 does is a tumor suppressor. What will happen is if the DNA is damaged, because you don't want to pass on any of that damaged DNA, this transcription factor gets activated, and what it transcribes is P21. P21 is an inhibitor of either the cyclin A CDK2 complex or an inhibitor of the cyclin E CDK2 complex. So it'll prevent the cell from moving forward through the cell cycle by blocking either cyclin E CDK2 activity or cyclin A CDK2 activity. I've also got here in the notes that one of the ways that you inhibit these inhibitors is what will happen is cyclin E or A bound to CDK2 can phosphorylate these family of proteins and mark them for degradation. So that is a negative feedback loop. So these guys can actually eventually inhibit their inhibitors, okay? And then you guys know about WEE1, that's the one that's gonna target CDK1, and it phosphorylates it, it puts an inhibitory phosphate on a CDK1. And we talked about this before, that CDC20 P25 is the phosphatase that takes off that inhibitory phosphate so that CDK1 can be active during its appropriate phases of the cell cycle. We also talked about last week the fact that cyclin B, when it binds to CDK1, it blocks WEE1 activity, and cyclin A, CDK1 does that as well. Okay, so participating in a negative feedback loop to block the inhibitor. Okay? All right, so here's some evidence, experimental evidence of the ecm4 family. So that was the first family that I showed on the slide, I was telling you that the levels of that protein are high during G0 phase, so that protein sequesters CDK4 and CDK6 away from cyclin D, so that you can't form this complex, so that the cell can't make it through G1. Okay. So here, what this is showing is a protein blot or a Western blot, and they probe for the ecm4 family member P16. And these are, they collected different types of cells. So these are cells that are super proliferative, and these are cells when they synchronize the cells. And these are cells that are hanging out in G0, so quiescent state. And what you can see here is these quiescent cells have high levels of that P16 protein, okay? So this is experimental evidence that in proliferating cells, you don't have the P16 or ink family proteins, and in quiescent state, those proteins are present. Okay. So I was telling you before, so that's the ink family. I was telling you before P27, its levels are high during G0, so it's an inhibitor of CDK2 activity. And here you can see when you add in platelet-derived growth factor, so here these are cells without any mitogen, and then you add in platelet-derived growth factor and you see that P27 levels go away. So again, this is evidence that once the cell has entered the cell cycle, P27 levels are going to be downregulated so that you release any inhibitory activity on the CDK2. And this is just showing the signaling pathway. So we talked before about how the platelet-derived growth factor and these mitogens signal through these receptor tyrosine kinases. And here, that you've seen this blot before, so here this is when they took cells and they either overexpressed the PI3K AKT pathway, or they overexpressed the ERK pathway. And you can see here that when you overexpress these pathways, you downregulate KIP protein levels, okay? And of course, we see cyclin D go up because we know that cyclin D is induced by the mitogens. So this is again showing you through this mitogen signaling pathway, you're down-regulating those KIP proteins. And then you actually see that here, right? So these pathways block KIP, P27, and that allows the cell to go into the cell cycle because P27 normally blocks cyclin D. Blocks CDK2. Okay. So now, if there was no DNA damage, the cells would be able to proceed. But if we do have DNA damage, I was mentioning before that this protein called P53 is a transcription factor. It's mutated in about 50% of cancer. If there is DNA damage during the early phases of the cell cycle, what's going to happen is P53 gets activated, this transcription factor, it'll translocate into the nucleus, and it'll induce transcription of P21. And P21, again, is this inhibitor. It's going to block the cyclin E-CDK2 activity so that the cell cycle can't move forward. And this is part of that G1, this is that G1S restriction point. So here is experimental evidence of that. Okay, so here what they did, they have this drug called DAP that damages DNA, okay? So they took some cells, they added DAP to the cells, and it ended up damaging their DNA, and they collected over time. So this is over 36 hours after the cells were given this DNA damaging treatment. And what you can see is that over time you get an increase in P21. The reason you're getting an increase in P21 protein over time is because the P53 transcription factor is activated and it's inducing transcription of the P21 gene. So now you have an increase in P21 protein. And what this is showing here, this is old school way, now we do qPCR. This is a northern blot, so this was a way back in the day that we used to look at RNA levels, look at transcript levels. Here you can see an increase in P21 RNA. So again, P21 is getting transcribed by P53 in response to DNA damage. Now, once that P21 is available, what it's gonna do is actually bind to CDK1, CDK2 or cyclin E. So these are immunoprecipitation, this is IPs, either for CDK2, and then they probed for P21, so they pulled CDK2 out of the lysate with an antibody, and then they ran that IP onto a Western blot, and here you see an increase in P21. So what this is telling you is that CDK2 and P21 are forming an interaction. And here they do the same thing, they IP for cyclin E, and they look and see if P21 is bound, and yes, it is. So P21 is binding to cyclin E in response to that DNA damage. So what this is telling you is that you have a complex forming between cyclin E, CDK2, cyclin E, and P21 in response to that DNA damage. Okay. And then finally, as I was telling you, this blocks the activity of cyclin E, CDK2, so that the cell cannot proceed through the cell cycle in response to DNA damage. So that's what's shown here. So they IP for CDK2, so this is going to be an in vitro kinase assay. So they IP for CDK2. They see that cyclin E is bound. They see that P21 is bound in response to DAP, so in response to that DNA damage. P21 is now binding to this complex, and now you don't get phosphorylation of the substrate. In this case, they use histone 1. Okay? So in the absence of DAP, if there's no DNA damage, cyclin E, CDK2 forms the active complex, and it can phosphorylate whatever substrate it is. In this case, they use histone H1. But if P21 is present in response to DNA damage, now you don't get phosphorylation of that substrate. That's because P21 is blocking the activity of the cyclin E CDK2 complex. All right, so if everything's good, then yes, the cell is going to proceed forward and enter into, M phase. However, we know we have the DNA, we have a checkpoint here, so if DNA damage happens here, we know that we can have P53 can actually block CDK1 activity. This is not through P21, this is through an indirect mechanism. Some proteins called 14-3-3 proteins will actually be activated through P53, and they inactivate CDK1. So it's not through P21, but P53 can block CDK1 activity in response to DNA damage. So again, so the cell doesn't enter into M phase. We know that CDC25 is involved in removing inhibitory phosphates. So you would want to, if there's DNA damage, you're going to want to prevent this from happening because you don't want active CDK1. So there's these proteins, these kinases called CHEK1 and CHEK2. And CHEK1 and CHEK2 inhibit CDC25 in response to DNA damage. So in response to DNA damage, CHEK1 and CHEK2 are, these are kinases that are going to block CDC25 activity so that you cannot remove that inhibitory phosphate off of CDK1, so that CDK1 stays inactive. And then of course we know that WEE1 can also come into play in response to DNA damage and put an inhibitory phosphate on the CDK1 to prevent its activity. Okay, so these are two ways you can block the cell, either through the restriction point here with P53 activating P21, or you can block at G2 if there's DNA damage through inhibiting CDC25, activating WEE1, and then P53 can also block. Okay. All right. And so if everything's good, you enter into M phase, and we talked about this last week. So if all is well, if you have proper alignment of your chromosomes, then the anaphase promoting complex is going to activate separase. Separase is going to cleave at the kinetochores, and then the two sets of chromosomes are going to be able to, one set will go into one daughter cell, the other set will go into the other daughter cell. The cell divides, and all is well. However, as we talked about last time, if there isn't proper alignment, then you have, that's where the spindle checkpoint is. So we went over this last time, but just to review it again, what happens is you have the complex here, this inhibitory complex. It's going to be actually, so these bug proteins along with CDC20 are going to be bound at the kinetochores. If there is proper alignment, or if it's proper alignment and if there's a kinetochore attached to each one, microtubule attached to each one of the kinetochores of the chromosomes, what's going to happen is these guys get released. CDC20 can now bind to APC. APC can then degrade to curin. Now separase is free to cleave the kinetochore so that one chromosome goes into one daughter cell and one goes into the other. As long as the kinetochores do not have, all of the kinetochores don't have microtubules attached, like right here, this complex is going to stay sitting at the kinetochores to prevent this activity from happening, prevent any separation. Okay. All right. Okay, you guys, that third quick question I didn't go over with you. So this one here, how does cyclin D accumulation affect ink-4 binding? What happens is as cyclin D starts to accumulate, it displaces ink-1. Okay? Do you guys have the questions written down? Yes? Okay. Okay, so Rachel, how does ink-4 maintain solubility 0?
So they're super high in G0, so they block cyclin D from binding to CDK4/6.
Yep. So the levels are high in G0. And so they're able to bind to the CDK4 and CDK6 so that it can't bind to cyclin D. And then what happens is, for the third question, what happens is as cyclin D levels start to rise, the cyclin D will be able to displace. It'll get to a critical mass where it'll start displacing the ink-4 levels. And then we also saw that the ink-4 levels go down, right? So the ink-4 levels start to go down in the proliferating cells. Coupled with that, cyclin D can displace, as the levels of cyclin D go up, it can displace binding to the ink-4 binding to the CDK4, CDK6. Okay. Can you answer what? Oh, I just said it. What happens to zinc levels when the cells exit G0, they go down? Can you tell us how is CDK2 activity blocked if DNA is damaged in S phase?
Yeah, so the protein P53 promotes the transcription of P21.
Yeah. And then P21 does what? It's gonna bind to what?
The CDK.
Yeah, so P21, so she's saying what happens is P53 gets activated, that transcription factor, it induces transcription of P21. P21 is a kinase inhibitor, so what it does is it's gonna bind to the cyclin E CDK2 complex and prevent that from having activity. And we know that based on that IP in vitro kinase assay, where they showed that if you have DNA damage, P21 levels go up. The P21 cyclin E CDK2 made a complex, and it prevented phosphorylation of the substrate histone H1. So cyclin E CDK2 activity was blocked by P21. Okay. Can you answer if CDK1 activity is blocked in, or how is CDK1 activity blocked if DNA is damaged or if synthesis is incomplete during G2? So what happens there in G2?
So in G2, if there's DNA damage, things like V1, V3, or CHK1/2, and then inhibit things that will inhibit the complex cyclin A to be in CDK.
So for the WEE1, how would it block CDK1 activity?
So V1 phosphorylates CDK, the cyclin A, I think.
It's gonna phosphorylate the CDK1.
Okay, yeah.
So just so you guys know, when you're talking about blocking activity, it's always going after the CDK, right? Because the CDK is the kinase. It's the thing that actually has enzymatic activity. So if you want to inhibit something, you're inhibiting the CDK. So that WEE1 is inhibiting the CDK1. How does it do it? How does it block its activity?
So it phosphorylates it. And
Yeah.
that, blocks like an active site.
Yeah, it's going to put an inhibitory phosphate on it. Okay, so you told us about the WEE1. Then you said the check1/2 proteins.
Yeah.
So they block which protein?
They
Which-
block CDC25.
And what does CDC25 normally do?
CDC25 normally unphosphorylates CDK1, which V1 phosphorylates initially, so it kind of blocks.
So that inhibitory phosphate that you were talking about that's put on by WEE1 on the CDK1, the CDC25, it's a phosphatase, so it takes it off. So you want to prevent, two ways of preventing CDK1 activity. One is to block with the WEE1, put that inhibitory phosphate, and then prevent CDC25 from coming along and taking it off with the check proteins. And then I was telling you P53, it's through some indirect mechanism, it also can block CDK1 activity. Okay. And then how is mitosis blocked if chromosomes aren't properly aligned? Who wants to answer that? So if the chromosomes aren't properly aligned, Joanne, if they're not properly aligned, how is mitosis blocked?
At the spindle checkpoint, the spindle assembly checkpoint, SAP is turned on and it inhibits APC and prevents chromosome misalignment. So it looks at if the kinetochores and the microtubules are properly connected to the kinetochores. If it's not, then BUB1 will connect to that complex and it will inactivate APC.
So the key there, the protein that is, so the BUB proteins are sitting there, but the important guy that's sitting there that's preventing the APC from being active is CDC20. So the CDC20 that's sitting there at that kinetochore, that guy needs to be released in order for it to bind to APC. Because remember, APC is the anaphase promoting complex. That's the proteasome. It's the E3 ubiquitin ligase that's going to put ubiquitins on a protein to mark it for degradation. And remember, CDC20 is the thing that recognizes substrates. Do y'all remember that when we talked about the proteasome last time? So the key here is that until all the microtubules are attached to the kinetochores, CDC20 is going to be sitting at the kinetochore along with those BUB proteins. Once all those microtubules are attached to the kinetochores, now CDC20 can be released, go off, bind to APC. The APC complex will then phosphorylate, I think it was securin, and then securin then releases separase. Separase can now go and cleave up the kinetochore, so now one protein can go, or one chromosome can go to one daughter cell, and the other chromosome can go to the other. The other thing to remember is that that APC complex also phosphorylates cyclin B and degrades it for marks it for degradation, so that you can actually exit mitosis. Okay. All right. So now we're going to move on. Are there any questions about cell cycle before we move on to cancer bio? Yes.
Didn't you say that cyclin B and CPA1 also like they block chromosome mitosis?
So what they will do is you want to be able to get out of mitosis. So you need to down-regulate cyclin B activity so the cell can exit. What I think you're referring to is when we're talking about chromosome alignment, when you knock out cyclin B. So there was that one where I showed if you siRNA'd or knocked out cyclin B, the chromosomes don't properly align. But now cyclin B does inhibit WEE1 activity. Is that what you were referring to?
I was referring to this type.
Let's see. That's the one I'm talking about. So that one, do you see at the bottom where it's on the second row where it says knock out cyclin B?
Yeah.
So that's getting rid of cyclin B. So if you get rid of cyclin B, the chromosomes can't properly align. So part of its role in getting the cell through M phase is for it to make sure, cyclin B is important in making sure that the chromosomes are properly aligned so that the cell can separate. But if you get rid of it, they don't properly align. Any other questions? Okay, let's move on to cancer bio. Biology lecture. Okay. So, cancer, this is a term, it's a disease in which cells divide without control. Okay, so they're dividing without any control, and this is very important, they're invading and spreading to nearby tissues. So that's what defines cancer. Cells that divide without any control, and they're going to invade and spread to other tissues. All right. So the issue with cancer is that it destroys healthy tissue, right? So you've got these cells that are proliferating, but the problem is once they spread and go to other parts of the body, they're gonna basically destroy any healthy tissue. So here's an example of a brain tumor. Here you can see the brain tumor. Here's the brain here, and you can see that it's pretty much taking over. It's destroying that healthy tissue. This is a normal bone scan. If you were to take a bone scan of a person that doesn't have cancer, this is prostate cancer here, where it's spread from the prostate to the bone, and you can see these really dark, imaging here. This is the cancer that's spread to all parts of the skeleton. So again, this is taking over that healthy, it's replacing that healthy bone marrow with cancer cells. Okay, here's another image looking at gross pathology here. You can see this is liver metastasis. This is from pancreatic cells. So here you can see that the tumors are taking over the liver, right? So again, just taking over that healthy tissue. This is what a person that has prostate cancer, the bone. So here's the bone marrow. It should be this red material here, but you can see all these white spots. That's the tumor cells taking over that healthy tissue. So the cancer cells, when they spread, they're destroying the healthy tissue. Here's also, we can stain, so this is a biopsy. So if a person has to get a core biopsy done, for example, what they go in is they can take some of the tissue, you could slice the tissue, put it onto slides, and look at the slides under a microscope and stain for biomarkers that indicate cancer. In this case, this is stained for the estrogen receptor. And here you can see this breast tissue. It's expressing a lot of the estrogen receptor, which is a biomarker for cancer. Again, you're losing the architecture of the normal breast. These cells are going to be destroying the healthy tissue, normal breast tissue. And so some of the consequences, some of the physiological consequences of cancer One is cachexia. This is responsible for about 1/3 of cancer deaths. So when I was showing you before, so you can see from these images here, you see how the tumor cells, this white part, is taking over that tissue. These tumor cells are highly metabolic, and I think Dr. Kim will talk to you guys about metabolism and that sort of thing in his section, but they're really metabolic, so they require a lot of energy. And so basically what happens is you end up losing skeletal and fat tissue when you have cancer because all that energy is getting sucked up by the cancer cells, okay? And the person can consume as many calories, it doesn't matter. They'll be pretty gaunt and lose a lot of muscle tissue. Another consequence, physiological consequence, is nerve damage due to neuropathy. So here, showing here, so this is common in prostate cancer where the man will get bone mets in the spine, and you can see here, this is a bone met right here, and it's pushing on the nerve. And so one of the symptoms of if a man has prostate cancer that spreads is they'll end up with a lot of back pain because the nerve is being pressed on. Another thing that happens as a consequence of cancer when it spreads is remodeling of the bone. So something that's common in breast cancer is osteolytic lesions where you end up getting lots of fractures. So that's what's shown here. This is an osteolytic lesion. When the breast cells get to the bone, they start just basically degrading the bone. And you end up with lots of fractures. Alternatively, in the case of prostate cancer, a lot of times it'll make what's called osteoblastic or lesions that build bone. And that's what's shown here. So here's an osteoblastic lesion where you're building bone, and again, that can press on the nerve. But you get a lot of bone remodeling that will mess with your skeleton and can end up with fractures and again pressing on nerves. And then of course, not surprisingly, you end up with organ failure when you have these tumor cells taking over healthy tissues. Okay? All right. So cancer rather is going to be classified by tissue origin. So we've got solid tumors, we have liquid tumors. So it's classified as a carcinoma if it's a cancer of epithelial cells. So an example for that would be breast or prostate cancer. Sarcomas if it's cancer of connective tissue. It's a lymphoma if it's a cancer of lymphatic systems. Leukemia if it's cancer of the bone marrow, and it's a myeloma if it's cancers of the plasma. And you see here that 80% of cancers are solid tumors, okay? And so they would fall under either carcinoma, sarcoma, or lymphoma. All right, so tumors can be benign or malignant, okay? So not all tumors are cancerous. So you could have a tumor, but it not be cancerous. So a benign tumor is non-cancerous. It's going to be it's benign because it hasn't breached the basement membrane, and I'll talk about that in just a second. A malignant tumor at this point, it is cancerous. So this is going to be tumor cells that invade nearby tissues or they spread to other parts of the body. So this is what defines whether something is cancer and whether or not it's malignant, okay? So again, not all tumors are actually cancer. So here's what, if you take epithelial cells, for example, so some sort of solid tumor, say prostate cancer, you've got normal epithelial cells. Right below our epithelial cells is something called a basement membrane, okay? So, and then this would be a blood vessel right here. This is your extracellular matrix, and then here is your blood vessel. Once these cells start growing, so let's say that this cell right here is gonna become tumorous, so it's gonna start dividing without control, right? So you've got all these cells dividing without control. At this point, it's not cancer. It's just a tumor, it's a benign tumor. What happens is, if these cells actually can break through that basement membrane, so they'll secrete proteins, if they're cancerous, they'll secrete proteins that can degrade that basement membrane. Once these cells break through the basement membrane, they can enter into the bloodstream and spread to other parts of the body. This is the point at which you've got cancer, at which point it's cancerous, okay? And then it can spread to other parts of the body, maybe it goes to the bone, maybe it goes to the liver, the pancreas, wherever, but then it's gonna spread to other parts of the body, colonize those other parts of the body, and that's what is gonna define cancer. Now, the important thing too here is you can also have, and I'll show you when we get to staging and grading, you can have this basement membrane breach, but it can still stay localized in the area of origin. So for example, you could have, say, prostate cancer, and it can, if it breaks through the basement membrane, it's still considered cancer. And if it spreads like local, we call that regional or local disease. If it spreads to, say, lymph nodes that are right nearby, that's considered cancer. But as long as it's contained and you don't have a breach of the basement membrane, it's considered a tumor and not cancerous. Okay. All right. So in terms of the spreading, the other thing that happens as well, the way that these cells are actually able to move, because technically along this epithelium here, these cells are, they're held in place, okay? So they're held in place because they bind to each other. They have connections that allow them, cell-cell connections that allow them to connect to each other, and they have cell basement membrane connections that allow them to stay put at the basement membrane. What happens is cancer cells will go from this epithelial phenotype, oh, here we go, so here's your cell-cell connections, here's your cell basement membrane connections. They'll go from being the epithelial phenotype to what's called a mesenchymal phenotype. So mesenchymal cells like to move. So they'll take on these characteristics of mesenchymal cells that then allow them to move and spread and crawl and go to other parts of the body, okay? So we call this EMT transition. So going from an epithelial to a mesenchymal phenotype. So these cells here, epithelial cells, have something called polarity. So basically that means the epithelial cell knows what's up, it knows what's down, it knows what's left and right. So this epithelial cell knows that this is the top, it knows that this is its side, we call this the basolateral side, and it knows that this is the basal side. It knows it's the basal side because this is the side that's connected to the basement membrane. In the case of losing cell polarity, these cells here don't have any of these connections, so they have no idea what's up, down, left, right. Okay, so they've lost their polarity. Another thing too here, this is what I was saying before, so these cells, these epithelial cells, they're gonna be attached to that basement membrane. These cells here are no longer attached to basement membrane, so they're gonna be able to crawl and move and spread to other parts of the body. Again, epithelial cells, they're stationary because they're attached. These guys aren't, so they're modal. These guys have high levels of a connection protein called ecadherin. So ecadherin keeps the cells connected to each other. These guys are gonna lose ecadherin, so they're no longer connected. And then another thing, these cells lose a protein called cadherin, and then they get high levels of it, and that again is indicative of the mesenchymal phenotype. So we can actually, in the lab, look to see if a cancer cell is going under EMT by looking for all of these characteristics and staining for proteins like ecadherin and seeing if they lose ecadherin. So here's an example of breast, this is breast tissue where you can see this is the basement membrane here in red. And then here you can see where these cells, these are now cancer cells, are going through EMT, see how they've breached that basement membrane, right? And now they're starting to spread. So this would be an indication. This is a normal breast duct here. So you've got your lumen, so that's what's here. You've got the epithelial cells, the layer of epithelial cells, so that's what's shown here. And then it's surrounded by this basement membrane that includes these myoepithelial cells. But now here, this is becoming cancerous. These guys have breached that membrane. All right. So we're talking about staging. We can stage the tumors based on disease progression. So in situ is when it's precancerous. So this is going to be that the cells are proliferating abnormally, but they're only in the layer of cells where they're developed. So at this point, if we go back here, okay, so if we go back here, if these cells hadn't breached, so if they started proliferating, this layer of cells started proliferating and you started getting a bunch of cells here, but they haven't breached the membrane at this point, it's gonna still be in situ, okay? It hasn't breached the membrane. Okay, now localized is gonna be, call it localized, that's gonna be called, okay, so in situ is either precancerous or you'll see the staging TIS. So the IS is in situ. So if it's localized, it gets a stage of either T1 or T2. So that's gonna be where it's breaking the basement membrane, but it's staying confined to that organ, okay? Regional is gonna either get a staging of T3 or T4. or it'll get an end stage. So this means that the cancer has spread beyond that primary site where it started, and it's gone to, say, lymph nodes, which are regional, which are nearby. Okay, so the N is for the lymph nodes. So here, this N0, you will get an N0 if it's starting to spread, it's breached the basement membrane, but it hasn't made it to the lymph nodes. Once it gets to the lymph node, you get an N1. And then once it goes to distant sites, so like if it was breast cancer, if it goes to the lung, if it's prostate cancer, if it goes to the bone, that's considered distant metastasis. So that's gonna get an M for metastasis and M1. If it hasn't metastasized, it's an M0, okay? So here is, just using breast cancer as an example, here is what a doctor would look for. So they would do this is a normal mammogram. Here would be a mammogram if a person had cancer, so you can see the cancer right there. And the way they would stage it is based on size and spread of the tumor. So here, this would be in situ. So this is gonna be, the tumor cells are starting to proliferate, but they haven't reached basement membrane. Once they see that tumor start to get just under 2 centimeters, that's gonna be considered T1, but it's still localized. T2, it's still localized, but it's now getting larger. And then once you hit T3 and T4, that's when it's starting to breach basement membrane and go to other parts. So here it's gonna be spreading to the lymph nodes underneath your arm. And then it would get an M rating if it metastasized to bone, if they found, or lung. Breast cancer normally goes to lung. So if they found tumors in your lung. Okay, so now you can also stage based on histology. So again, we would take the biopsy, you would take a biopsy from a patient, look at it under the microscope, and see what you see, okay? And so this is gonna be, what I'm gonna tell you now is the grading that we use for prostate cancer, and it's a Gleason score, but just real quick, what you look for when you're basing on a histology, you look for these 4 grades, okay? So this is not staging, this is grading. So you're looking to see if it's G1, that means it's low grade, that's well differentiated. What that means is if, for example, like the breast duct, if that duct still looks like a normal duct, that's well differentiated, that's low. It looks like what it's supposed to look like. G2 is when it starts to lose that ductal structure. And so it's moderately differentiated. G3 means it's starting to not look like a duct anymore, and I'll show you examples of this in a sec. That's poorly differentiated, so it's gonna be high grade. And then G4, completely undifferentiated, no evidence that there are even ducts ever there. That's gonna be the high grade, okay? So here is the grading system that we use for prostate cancer. It's called the Gleason, but here's the differentiation. So here you can see, this is differentiated, so you can see all the ducts, right? So these are prostate glands here. As it starts to get undifferentiated, if you compare this picture to this picture, this would be considered G4, right? So this is G1, G1, and this would be like a G4 here. So you can see that you've lost all the architecture of the gland, and now this is just this is like clearly cancer at this point, right? They've breached all basement membranes at this point, and you just got cells growing uncontrollably. So what we do here for prostate cancer is something called a Gleason score, the way we grade. So this would be here, so this would be differentiated. So what you do is you would, the pathologist, and this can be subjective, you guys. So they find that you could take 2 different pathologists, they might actually grade a tumor differently. That's why we can't just rely on grading. You have to also look at biomarkers, and there's other things that we're trying to do as well to be able to grade and stage people, because a lot of this can be subjective. So here, this would be get a Gleason grade of 2. This would get a Gleason grade of 3, 4, and 5. What you do in a given tumor is you look for the 2 patterns that are the most represented. So in a given tumor, you might get a mixture of, say, some of the tumor looks like this, and say some of the tumor looks like this. So you would add these 2 scores together, you'd add 3 plus 5, and that would give you a Gleason score of 8. Okay, so you look for the 2 most prevalent patterns. Now, if your entire tumor looks like this, then your Gleason score would be 4. 2 plus 2 would be 4. So you're adding up the 2 most prevalent patterns. Obviously, the higher the number, so if you end up with a 10, right, then that's gonna be a pretty high grade tumor. So anything, we pretty much consider anything sort of 7 and above is bad. 2, 3, Gleason scores of like 2, 3, that you're okay, okay? And so here's an example of putting together staging and grading. So here's an example. So this person could have a T4, N1, M0, and have a Gleason score of 7. So they had prevalent, when you looked at the tumor under the microscope, it looked mostly like 3 and 4. So this is gonna be a tumor that's spreading from the prostate into nearby tissue. So that's why it's a T1. It has spread to the lymph node, so that's why it gets an N1. It hasn't metastasized yet, so that's why it gets an M0. And it's showing primarily moderately and poorly differentiated cells in the biopsy, so that's why it's getting 3 and 4. Okay? And the significance, you guys, of this staging and grading is not just to diagnose you, but to also dictate what kind of treatment you get. Yes?
So when you have Gleason 7, do you also say you put 3 plus 4? As in for if you're actually like notating this?
You would, I don't know if the pathologists actually do the 3 plus 4. They may. They actually may. But when I've seen it, when I've generally seen it, they just give you just the Gleason score.
Okay.
Yeah.
You could be like 5 and 2. Just
You could be 5 and 2, but let's say you're 5 and 2, this is still really bad.
Yeah.
So even if you were, whatever combination makes up 7 is not gonna be good for you, right? Yeah. Okay. Okay. Okay, do you guys have the questions written down? Do you guys have them all written down? Okay, so what are some of the and all of these, you guys know you can just go to the slides and the answers are in the slides. So what are some of the physiological effects of cancer? Can someone just answer that? physiological effects of cancer?
Cachexia,
Yeah,
peripheral neuropathy, bone remodel, or
yeah,
peripheral remodeling, and arthroplasty.
yes. Okay. Define the stages of cancer. So you can just give me the names of them. You don't have to tell me what all they are.
So we have in situ, localized, regional, and distant.
Yes. Okay, grades of cancer? Grades of cancer? Do you know the grade, can you define the grades of cancer? Up there. That's looking, that's turning his head. Yeah,
Sure, the grades of cancer is going to be G1, G2, G3, and G4. G1 would be well differentiated, meaning you still have structure. G2 would be moderately differentiated, where you would still have somewhat semblance of the structure. Even less so in G3, where structure is kind of disappeared, but you can still maybe see something. G4 is just a mass of cells with uncontrolled growth, with everything else. There is no structure or function.
right, and at that point, your basement membrane is gone at that point, yeah. Difference between tumor and cancer, gray shirt. Difference between tumor and cancer. And then also difference between benign and malignant. So you can have a tumor, it doesn't have to be cancerous. So actually, let's go to what makes something cancer. So what would make it cancerous?
Something that, when it's cancerous, has the ability to spread.
Yes. So the key is when it's cancerous, it can break through the basement membrane and spread. And so that goes back to the difference between benign and malignant. So it's benign if it doesn't spread and it stays contained. It's malignant at the point that now it breaks the basement membrane and it can spread. Okay? And then what are the types of cancers based on tissue origin? You just give me the names. That was that slide where it had the sarcomas and so what are the types? Nikhil? Do you have your slides? Yeah, so, yeah, just, yeah, so those fell under solid. Then there's also gonna be the liquid tumors and the myelomas, yeah. And most of the cancers that we deal with are the solid tumors. About 80% are gonna be the solid tumors. And then Nikhil, can you answer this one? So which cancer is gonna be more aggressive and why? The T4N0M0 with a Gleason score of 7 or T4N1M1 with a Gleason score of 7?
It'd be the second one.
Yeah, and so why is that?
I remember somebody do like the white grade.
So it's not, so they both have the same grade. So that Gleason score is the grade. It has to do with the staging in this point. So what's the difference between the staging? So they both have the same Gleason grade. They're both 7. So when you looked at that tumor, primary tumor under the microscope, they had the same kind of architecture. But at this point, this person not only has tumor cells that are in the prostate, for example, now you got tumor cells that have spread. So that's the N and the M1. The thing, you guys, just to make a point here about the staging versus the grading. So the grading, the idea behind the grading is you can actually access the tumor to take it out of the person to look at it under the microscope. In the case of staging, you may not actually get access to that tumor, right? So like if something had gone to the bone, you're not gonna do a bone biopsy on the person. Bone biopsies are incredibly painful, and so you're not gonna put somebody under that type of pain to get the bone biopsy out to look at it. So at that point, we know once it's gone to the bone, at this point it's malignant cancer, right? So there's no sense in putting a person under that kind of pain just to take it out and look at it. A lot of times when things have metastasized, the images that we get or the histology we're able to get is after the person has passed away. So during autopsy, they can get the bone or get the metastatic tissue. Okay, so the staging is going to tell you how big this tumor is, has it spread. The grading is telling you the architecture of the tumor. We call it the primary tumor, the tumor that's localized to where it starts from, whether it's a breast tumor and it's localized still to the breast, and we can take that tumor out and look at it under the microscope. Okay, so in this case, the prostate tumor had the same, that they were able to access from the prostate, had the same Gleason score, but then when they were staging this person, they probably did a bone scan on this person and saw, this one person, they had mets in the bone, so that's gonna be a more aggressive cancer, okay? All right, so in the last couple of minutes, because I wanna try to get through the next few slides, these are the hallmarks of cancer. Dr. Kim might talk about this too in his part of the lecture, but the hallmarks of cancer, this is the Bible for cancer researchers, okay? So there are these hallmarks. So this paper here, this Hanahan-Weinberg paper, I think has been cited, oh, I don't have it in my notes, let me see. Dang it, it's been cited like maybe 20,000 times.
You had it in the next.
I did, I had it on the next slide, okay.
Yeah.
Oh, here we go. Okay, so it's been cited more than 18,000 times. You guys, it's a lot of citations. Lots and lots and lots of citations. I'm happy if I have like 13 citations on my paper. So this is a big deal. So these are the hallmarks of cancer. Let me just go back. What they are is there's commonalities between cancers, these different cancer types. There's commonalities between breast and prostate and ovarian and pancreatic, and so they all have these interconnected characteristics. And the goal of cancer research is to understand the how. So how are these cells, these normal cells, taking on these hallmarks, and why? So how and why, because then that way, if we know the how and the why, we can circumvent them. So these are going to be the hallmarks. So you've got invading growth suppressors. So those things that prevent a cell from dividing uncontrollably, cancer cells could care less. They're going to divide anyway. Avoiding destruction of the immune system. So actually, we think of a cancer cell as a wound that will not heal. So they're actually, when you get, say, a solid tumor, immune cells actually will try to attack that tumor and make it go away. The problem is cancer cells take advantage of those immune cells to actually survive. Enabling replicative immortality. So what this has to do with is on the ends of your chromosomes are things called telomeres. And telomeres help when you're replicating your DNA so that you don't shorten the DNA over time. You have these overhangs. And what happens is as we get older, our overhangs get shorter and shorter and shorter. And eventually the cell senesces and you pass away, right? So telomeres, the problem here is that cancer cells have a telomerase that keeps adding the telomeres. So it keeps the cell from being able to replicate pretty much indefinitely because the telomeres don't shorten. Tumor promoting inflammation, so that has to do with this avoiding immune destruction. These immune cells bring in inflammatory cytokines into the tumor microenvironment, and technically these inflammatory cytokines should kill the cells. Cancer cells don't care. They actually use these inflammatory cytokines to grow and proliferate and survive. This one is activating invasion and metastasis. So this goes back to that EMT, where the cells go from being epithelial to mesenchymal-like, and so now they can move and breach the basement membrane and move to other sites. We've got inducing angiogenesis. So this has to do, you guys are reading a paper right now, and it talks about angiogenesis. So this is when the tumor cells can promote the formation, can activate endothelial cells and promote the formation of blood vessels in the tumor to feed the tumor. Genomic instability and mutation. So here, remember in a normal cell cycle, when the DNA is damaged, the cell is going to say, look, I'm not going to divide anymore. In the case of a cancer cell, remember P53 is mutated. So in the case of a cancer cell, it doesn't care. If the DNA is damaged, it's going to pass on that mutated DNA anyway. And then, let's see, resisting cell death. So here, this is again has to do with P53. One of the things I didn't tell you guys that P53 will do is promote apoptosis of the cell. So it'll block the cell cycle and give the cell actually time to repair its DNA before moving forward. But if the repair is not repairable, if the DNA damage is not repairable, the cell will die. Cancer cells don't care, they're gonna live anyway. And then deregulating cellular energetics. This is Dr. Kim's, his area of expertise. And so this has to do with metabolism and cancer cells being able to use metabolism to survive. Yes?
Is that what basically results in, I'm sorry, I find the exact term here, the like muscle degeneration?
Yeah.
Okay.
Yeah. Okay, so these are all the, oh, and then I don't know if I did this one, sustaining proliferative signaling. Oh, So here, this one's really important. So remember how, like, we talked about platelet-derived growth factor and mitogens promote cell cycle entry? The thing with cancer cells is they will be able to proliferate whether or not you give them signals, external signals to proliferate. So they'll either they'll make their own cytokines and promote their own growth, or they'll have what we call constitutively active pathways that they don't care if you have a signal there or not. The next signal, they're just going to be continuously active. Those those pathways downstream of the receptors are going to be continually active. Okay, so these are the hallmarks of cancer. All right, and so the tumor microenvironment, I just want to talk about this real quick. So this is what I think I've shown you guys this picture before. So in the tumor itself, so even when you do a biopsy of a tumor and you're looking at histology of the tumor, it's not just cancer cells that are in the tumor. You're going to have blood vessels that are invading into that tumor. So you've got these endothelial cells that make up blood vessels. You're going to have fibroblasts that are part of that tumor. Remember I was saying that a tumor is a wound that doesn't heal, so your body is going to try to fight it by attracting immune cells. So cells that are produced by bone marrow, it's going to attract those immune cells to the tumor microenvironment. The problem is all of these cells, these fibroblasts, these endothelial cells, the immune cells, all of these things sitting here in the tumor through paracrine signaling are going to promote growth and proliferation of those tumor cells, okay, and promote spread. Okay, here is an image this is an image of a pancreatic cancer, so this is a biopsy here, and what they did is they immunostained, so this is just more of the point. The red cells are the tumor cells, and then here you can see in this biopsy, in addition to those red pancreatic cancer cells, you also have, the tumor cells are in blue. The tumor cells are in blue, and then in addition to that, you have the immune cells that are in red, so you can see all this immune infiltrate into the tumor, and then the fibroblast cells are in green. So again, this tumor microenvironment has a bunch of different cell types. They're all communicating with each other. They're all secreting factors that are going to promote the growth and the proliferation and the survival of the cancer cells. Okay, and then as I talked about before, these cancer cells, they divide without control, so that goes to the cell cycle, which you guys are aware of. And this is just the people that got, actually I didn't mention this when we talked about cell cycle, but they got the Nobel Prize for defining the cell cycle. And here is P53. So remember I was telling you guys before, we know in response to DNA damage, P53 goes up or gets activated, you get P21 transcribed. P21 is going to block cyclin ECDK2 activity. If the cell is able to repair the DNA, then, so P53 will arrest the cell cycle in response to DNA repair. If the cell is able to repair the DNA, then the cell cycle is going to restart. If it doesn't repair it, the cell should die through cell death. Cancer cells, that doesn't happen. And so what you end up with is genetic instability. That's part of that hallmarks of cancer. So let me just show you an example of that. So remember we had talked about before, we talked about this whole idea, we know that we've got interphase, and then here these are the cells going through mitosis. We know we have to have chromosome duplication. The chromosomes go, centrosome duplication. The centrosomes go to either pole, and then the centrosomes are going to radiate off these microtubules that are going to bind to the chromosomes at the metaphase plate. So each pair of the chromosomes, one microtubule is going to bind to one kinetochore, the other microtubule is going to bind to the other one. It'll pull them apart during cytokinesis. So centrosome duplication is really, really important. Here's what happens if you have screwed up centrosome duplication. So this is correct centrosome duplication. You get one pair of centrosomes are going to be on one side of the pole of the cell. The other set are going to be on the other side. You get equal division of the chromosomes. If the centrosome duplication is screwed up, so if you make more than what you need, then you end up with what's called aneuploidy. So instead of getting an even division of the chromosomes, you're going to now, in the case of this cell, you're going to have, some cells might have extra chromosomes, some cells may not have enough, because basically it split the cell into three, right? So here you've got three pairs of centrosomes. They're all radiating out microtubules that are binding to kinetochores, and so you're basically pulling these chromosomes, instead of pulling them in an even direction, now you've got pulling them in multiple different directions, okay? And so you can either have this occur where you end up with cells that have are gonna have, less chromosomes. Here, you can also have, where you've got, in this case, when you over duplicate it, they've got 3 sets of centrosomes over here, 2 sets over here, so these guys are radiating out more microtubules, so they're gonna be able to pull some extra chromosomes over to that cell. So this cell has less chromosomes, this cell has extra. In any case, if you don't end up with the correct number of chromosomes, that's called genomic instability. The problem in cancer is if this guy, for example, let's say this guy right here was actually a tumor suppressor. Now what you've ended up with is a cell that has lost a tumor suppressor. If, for example, this is an oncogene, now you've ended up with a cell that has an oncogene that's promoting cancer. So that's part of the problem with this genomic instability, is moving chromosomes that could then ultimately end up with the cells dividing without control. Okay, so P53, here's some evidence, experimental evidence of P53 being involved in promoting genomic stability. So these are prostate cancer cell lines. This prostate cancer cell line has normal P53. These 2 here have either mutant P53, so P53 that doesn't function, or doesn't have P53 at all. Okay, so this is the other cell line that doesn't have it at all, and this is the Western blot showing this guy doesn't have it. So what this group did is they looked at the dividing cells under the microscope. So here is normal division occurring. Here's one set of centrosomes, here's the other set of centrosomes, here's the DNA being pulled. One half of the chromosomes being pulled to one daughter cell, the other half is being pulled to the other. In this case, if you have amplification of the centrosomes, so if the DNA, if the duplication doesn't happen correctly, now look, you've got this the chromosomes being pulled in 3 different directions. So the ultimate daughter cells that you end up with here are not going to have the correct number of chromosomes. And what they're showing here, they just measured it, so they showed in the LNCAP prostate cancer cells that have normal P53, most of the cells, 65% of the cells, had a correct number of centrosomes. So one pair here, one pair here. Look here with the D145 and PC3, they have amplified numbers of centrosomes. So that again was leading to this aberrant centrosome, aberrant division where the chromosomes are not gonna, you're not gonna have the right number of chromosomes in the daughter cells, okay? And then here, okay, here, let me see. Oh, okay, so that was the point I was making there, okay. And then here, let me, wait a minute. Oh, no, this is the point here. So what they did here, remember that PC3 cell line doesn't have P53? In this experiment, what they did is they added P53 back into that PC3 cell. So they gave it a wild type P53, normal P53, and here you see you end up with less cells having these 3 pairs of centrosomes. So they were able to rescue this phenotype, make the cells behave more normally by putting P53 in. So again, this is evidence that P53 is leading to genomic instability, because P53, when you have, and we don't know the exact mechanism of why P53 promotes correct centrosome amplification, but this is evidence that it has a role in that. Okay, and then this will be the final slide. So this one's actually really cool phenomenon. This happens in breast cancer. A group discovered this some years ago, and it's called intosis. What happens is, this is when a cell engulfs another whole cell, okay? And then, so here, this is so breast cancer does this a lot. So here's a breast cancer cell. It's engulfed another breast cancer cell. So there's the nucleus. This guy here that engulfed that other one is actually trying to undergo cell division. So it's replicated its DNA. So there's one DNA, set of DNA replicated. The other set, it's trying to undergo cytokinesis, but it can't because this guy is physically in the way. So you end up with a cell that's got 3 sets of chromosomes, right? Which is not the correct number, right? You want 1 set of chromosomes. And so that's this phenomenon here in intosis, and this is just showing fluorescence imaging of that. So here's a cell, it engulfed another cell, it's trying its best to divide, it can't because this guy is physically in the way, so now the cell ends up with 1, 2, 3 sets of chromosomes, okay? So this is another way that you can end up with genomic instability. All right, we're gonna stop there. I think I'm on